Monsanto, along with a handful of other giant corporations like DuPont, Dow, BASF, Bayer, and Syngenta, have taken over the food supply with genetically modified organisms, GMOs, that are bad for human health. Scientist Nancy Swanson has compiled information on the dangers of GMOs, showing graphs of its correlation with many rising cancers and diseases in the document entitled Genetically Modified Organisms and the Deterioration of Health in the United States, 2013. Tobacco plants were first genetically engineered plants modified to resist antibiotics in 1983 in France. They were later modified to resist herbicides and then to resist insects by producing its own insecticide. In 1995, the FDA approved all genetically engineered soy, cotton, canola, potato, squash, and tomato for commercialization. Later, corn, papaya, alfalfa, sugar beets, apples, peas, and rice would be added to the list. No safety tests by the FDA are performed under two false beliefs. One, that genetic engineering is no different than hybridization of crops practiced for centuries, and two, that non-GMOs are equivalent to GMOs. According to this paper in the International Journal of Molecular Sciences, it states, substantial equivalence concept is based on the assumption that commercialized traditional crops have been consumed for decades and have gained a history of safe use. Therefore, they can be used as comparators for the safety assessment of new GMOs derived from established plant varieties. It makes no sense that a non-GMO plant would be the same as a GMO plant. Then it goes on to at least admit that one of the central safety issues under debate regarding GMOs is the occurrence of unintended changes resulting from genetic transformation. Mutation surely brings unintended, unknown, and uncontrollable consequences and mutations. Plants have more than 45,000 genes, and scientists don't know what each gene does or how it interacts or is dependent to other genes. There are changes that aren't visible or detectable. It's all shooting in the dark science. Virtually all crops are genetically modified crops in the United States. According to the FDA, since 2018, 94% of all soybean, 94% of all cotton, 92% of all corn is genetically engineered in the United States. In 2013, GMO canola made up 95%, while sugar beets 99.9%. Bacterial transformation is a common genetic engineering process in which fragments of bacterial DNA are inserted into the DNA genome of plant cells. Some of the seeds of the transgenic plant will contain the transgenes. These foreign gene expressions changes many things about the organism, its environment, and has effects upon our bodies. Genes produce amino acids, which form proteins, and thus with genetic modifications, plants produce unknown proteins, which are xenobiotics, foreign substances to nature. Foreign unnatural substances trigger the immune system, causing allergic reactions, chronic inflammation, and autoimmune diseases. Rheumatoid arthritis is an immune response that has increased in correlation to the increase of GMO crops, in the Swanson report. GMO crops are modified to either create pesticides or resist pesticides. There are two main kinds of pesticides. One, insecticides that repel, kill, or control animals and insects. Two, herbicides that kill undesirable plants and weeds. There are three main purposes of gene modification. One, to make the plant produce its own insecticide that kills bugs when they attempt to eat the plant. Two, to make the plant immune from being harmed or killed by toxic chemicals and herbicides when sprayed on them. And three, to improve the plant phenotype, which is not related to pesticides. The third type is less common. Giving a plant a so-called positive trait is usually accompanied with an obvious negative trade-off. For example, the GMO potato was modified through gene silencing in 2015 to not discolor when bruised, merely an aesthetic change. The scientist Dr. Klaus Romans, who developed these potatoes, renounced his work and wrote the book Pandora's Potatoes, The Worst GMOs. He said, quote, most GMO varieties were stunted, chlorotic, mutated, or sterile, and many of them died quickly, like premature born babies. The genetic modification silencing the melanin gene creates more alpha aminoadipate, a neurotoxin. While the potato doesn't appear to bruise, you no longer have a visual indicator to tell you whether it is going bad and can eat the toxin tyranny. The same kind of genetically modified trait change is happening with the GMO apple. It would be much more productive to work on improving the terrain, like increasing mineral content of the soil and not using toxins at all in the farming process. Not only are specific unnatural genes inserted from one organism to another, but other chemical configurations must be made, such as removing introns, transferring promoters, and inserting terminator sequences. It's a messy process. Let's talk about insecticides. Bt toxin comes from Bacillus thuringiensis. In natural form, it is a soil-borne bacteria that makes some proteins toxic to certain insects. Pesticide products have been made using this bacteria. With genetically modified Bt crops, Bt genes that create protein toxins or insecticides are inserted into the plant's genome, changing it so that every cell of the plant manufactures this insecticide. This way, no insecticidal spray needs to be used, as the plant will kill off bugs itself. Common Bt crops are corn, cotton, and soybeans. This article claims that 90% of cotton planted in the USA, India, China, Australia, and South Africa have Bt genes. In 1995, Dr. Arpad Pusztai was hired by the European government to create the protocols to test the safety of GMOs. One of his experiments tested the genetically engineered potatoes that produce the Bt toxin with a snowdrop lectin gene on rats. And even though testing on animals is immoral, since these experiments were already done, we should heed their results. Also knowing that if we have to test food on animals in the first place, we are doing the wrong things to begin with. In Pusztai's experiment, there were 3 groups, one that ate non-GMO potatoes, one that ate Bt GMO potatoes, and one that ate non-GMO potatoes spiked with the natural Bt insecticide. He discovered that even though the non-GMO spiked with Bt toxin didn't produce negative effects on rats, the GMO potato did, revealing that the synthetically produced Bt toxin was different and that there were harmful byproducts of the engineering process itself. Rats that ate GMO with the Bt gene endured damage to the intestinal walls and had decreased size of the brain, liver, and testicles. There was also impaired development of the thymus, spleen, and immune system compared to the control group that did not eat GMO. After the study, Pusztai was ostracized from the scientific community, suspended from work, censored, and banned from speaking publicly. He was forced to retire. While GMO advocates claim that the Bt toxin is harmless to other non-insecticidal species, including humans, they are talking about the natural form. But the unnatural form is not equivalent, a fundamental error of the entire GMO ideology. The article entitled, "Have Monsanto and the Biotech Industry Turned Natural Bt Pesticides into GMO Supertoxins?" claims. Whereas natural Bt toxins are insoluble crystals with complex structures built around a DNA molecule, all GMO Bt toxins are soluble proteins with no DNA. GMO Bt toxins often have added to them synthetic or unrelated protein molecules. Thus, the absence of DNA in genetically modified Bt toxins reveals that there is no intelligent code in these proteins that manages triggers and timing of toxicological release, as the bacteria releases these toxins much more selectively and purposefully than the synthetic version. The article states, "The key inference from this understanding is that GMO developers, by solubilizing or shortening Bt toxins, have removed some or all of the inhibitory structures that make natural versions safe for most organisms." In this article, it claims that a review of biosafety application documents for 23 globally traded Bt pesticidal GMO crops events, as well as peer-reviewed research and patents, was conducted to compare GMO Bt proteins with natural ones. The study found 6 types of differences, both intentional and inadvertent in origin. This included finding that GMO crop plants themselves causes changes to the nature of Bt toxins. Not a single one of the 23 Bt commercial lines analyzed was identical to natural or historically used versions of Bt toxins. Natural Bt toxins are large, insoluble, and non-toxic precursors requiring unusual chemical conditions to become active toxins, but the processing undergone by all GMO Bt proteins typically causes them to be more toxic. Worse, they cause them to be active against many more species than natural forms of Bt toxins. Thus, Bt developers have been commercializing pesticide-containing GM crops with increased and broadened toxicity as compared to the normal limited toxicity ranges of natural Bt toxins. The researchers categorically state that any references to any GMO Bt toxins being natural are incorrect and scientifically unsupportable. Genetically engineered Bt kills insects by producing crystal proteins that bind and poke holes in an insect's guts until it dies. So what does this toxin do to human guts? In 1998, a study was performed on mice fed on GM-Bt toxins, and electron microscopy revealed degenerative forms of mitochondria, endoplasmic reticulum, and short microvilla. In 2012, the scientific report entitled Cytotoxicity on Human Cells of Insecticidal Toxins, etc., indicated that Bt toxins from GM plants can damage human intestinal walls. Damaged intestinal walls absorb less nutrients. Since the advent of GMOs has been the rise of gastrointestinal issues like gluten disorders, impaired digestion, damage to the intestinal wall, and leaky gut syndrome, which is when damage is so bad that the toxins and incompletely digested proteins, fats, and wastes leak into the bloodstream. This triggers an autoimmune reaction, which leads to abdominal bleeding, excessive gas and cramps, fatigue, food sensitivities, joint pain, skin rashes, and autoimmunity. Swanson shows the correlation between inflammatory bowel disease, chronic constipation, functional intestinal disorders, and deaths due to intestinal infection with the increase of Bt crops. According to this description on Bt, insects have developed immunity to the genetically modified Bt-containing corn when the GMO corn has, against best agricultural practice, been planted in the same plot year after year. This reveals that our toxic plants may not even serve a purpose anymore. We are told that Bt toxins pass through the body, which is a lie. In reproductive toxicology, these proteins were found in the blood of pregnant women and fetuses. In 2012, a study led by Giles Eric Cirilani linked GMO corn to rat tumors. The study was retracted by the publisher against the author's objections, who claimed the retraction was made by a Monsanto lobbyist and was a public health scandal. Crops are not only modified to create their own insecticide like Bt, they are also given an herbicide-resistant gene. Monsanto's Roundup is an herbicide that kills undesirable plants with glyphosate and other toxic chemicals. Their crops cannot be harmed by glyphosate because they have a glyphosate-resistant gene. Approximately 80% of crops are modified with this gene, which means 20% have the insecticidal gene. Giving crops both types of modification is called gene stacking, which started in 1995 with cotton. This NIH article about rice modification states, currently commercial transgenic crops are usually stacked with insect-resistant and herbicide-tolerant genes, as both traits are highly valuable in the production of major crops such as corn and cotton. Multiple insect-resistant genes and herbicide-tolerant genes were stacked in newly developed commercial transgenic crops. For example, a total of 8 genes for insect-resistant or herbicide tolerance were stacked in these trademark corn seeds released by Monsanto. According to this article in 2014, cotton and corn grown in the U.S. have both types of modifications, while sugar beet, soy, and canola have only the herbicide-resistant gene. The fact that corn and cotton have both types of genetic modification involving multiple genes for each indicates more dangers from unknown synergistic effects in the combination of these changes. GMO corn is found in the ingredients of corn flour, cornmeal, cornstarch, corn syrup, corn-based sweeteners, and flavorings. Pause this to read more hidden GMO ingredients. Many brands use genetically modified cottonseed oil, which doesn't even come from an edible plant. Wearing cotton means that pesticides and xenobiotics can make their way to our skin, which can eventually get into the bloodstream. While insecticide use has decreased, the use of Roundup Ready has greatly increased, so that the net use of pesticides has risen astronomically since the creation of GMOs. In general, pesticides have been linked to cancer, Alzheimer's, and Parkinson's disease. This article estimates that since 1974, over 3,520,000,000 pounds of Roundup have been applied. In 2015, Monsanto made $4.7 billion from Roundup alone. Also in 2015, the WHO described glyphosate as probably carcinogenic to humans. Glyphosate was first marked in 1976 and increased use over 6,500% from 1991 to 2010. And now it is detected in water, rain, and air, claims a 2011 geological survey. Glyphosate, which cannot be washed off and is absorbed into the food, is a known endocrine disruptor. This was shown in a 2009 article by Gasnier in Toxicology. In the report by Swanson, it says that the endocrine disrupting properties of glyphosate can lead to neurological disorders, learning disabilities, ADHD, autism, dementia, Alzheimer's, schizophrenia, and bipolar. Here are some graphs showing a correlation between glyphosate increase and the increase of these neurological disorders. As an endocrine disruptor, glyphosate affects mood through thyroid dysfunction. Rats and farm animals have exhibited uncharacteristic aggressive and antisocial behavior being fed GMO soy or corn. Since GMOs has also been an increase in thyroid cancer. Not only does glyphosate kill weeds, but also gut bacteria. This gut bacteria is required for the manufacture of tryptophan and other amino acids. Tryptophan is a precursor for serotonin, which controls insulin secretion. This absence leads to diabetes, and there is a correlation between the rise of diabetes and the rise of GMOs. Glyphosate is also known to disrupt the gut microbe by inhibiting the Shikimate pathway, claims a 2009 study in rats. This pathway is responsible for the synthesis of aromatic amino acids that are vital for the production of proteins, the building blocks of life. The endocrine disrupting properties of glyphosate can lead to reproductive problems like infertility, miscarriage, birth defects, and problems with sexual development. Glyphosate has been found in the breast milk of nursing moms. And let us not forget the issues of the genetic engineering process involved with glyphosate-resistant genes. A 2016 report found that in Roundup-tolerant maize, 117 proteins and 91 metabolites were altered in the metabolic pathway. Here is an image of all that is altered in the transformation process. This includes the production of putrescine and cadaverine, compounds that create the odor of death and break down amino acids in a dead body. In GMO crops, the alteration of these proteins causes imbalances in metabolism and increased oxidative stress. When looking at the link between GMOs and disease, we have to keep in mind that similar issues can be caused by both gene modification and from pesticides. Here is a picture summary from a scientific article of what we have so far discussed.
There are two major traits that have been genetically engineered. There's the poison drinkers and the poison producers. The poison drinkers are crops that are engineered not to die when sprayed with a normally deadly herbicide. Now, there's only been one human feeding study ever published on GMOs. It was done with the soybeans. And they found that part of the gene that is inserted into the soy to make that soybean Roundup ready, so it won't die when sprayed with Roundup herbicide, that gene actually transferred into the DNA of the bacteria living inside our intestines and continued to function. These folks had Roundup ready gut bacteria. So that means long after we stop eating this genetically modified soy, we may still have these genetically modified proteins produced continuously inside of us. There's also the pesticide producers. They're called Bt because the pesticide that they produce comes from a soil bacterium called Bt, or Bacillus thuringiensis. Peer-reviewed published studies show that when the Bt spray was sprayed over Washington State and Vancouver to kill gypsy moths, about 500 people reported allergy and flu-like symptoms. So what did the genetic engineers do? They took the gene that produces the toxin and they put it into the corn and cotton plants, where it produces this Bt toxin at thousands of times the concentration as the spray form. Although it's never been studied, what happens if the gene that produces the Bt toxin transfers to the bacteria living inside your intestines, like it happened with the soy? If that happens, it might turn our intestinal bacteria into living pesticide factories, continuously producing this Bt toxin inside of our intestines. What I say is, let's not be part of this dangerous experiment. Let's be careful to avoid eating the genetically modified foods, especially for our children who are most sensitive. Go to non-GMOshoppingguide.com for a list of non-genetically modified foods, or download the iPhone application Shop No GMO.
In addition to toxicity to humans, GMOs offer less nutritional value as they have depleted levels of mineral micronutrients. Not only are humans negatively affected, but the ecosystem at large. Herbicides destroy important microbes in the soil, pollute the air and water, and harm wildlife. Birds, butterflies, bees, and other species are dying off. Bees play a significant role in the pollination of the foods we eat. According to this article, in China, the native bee population is so low that farmers had to hand-pollinate their crops. Furthermore, the engineered genes spread throughout the gene pool and horizontal gene transfer, so that the damage done cannot be contained. In 2017, the government passed the National Genetically Modified Foods Labeling, which is supposed to be implemented in January 2022. Here's what the labeling looks like. Hopefully this will happen, but we all know whose side the government is on.